Thank you so much, Dylan. Thank you so much to all the organizer, to Irene, and to all the staffs that were very kind in assisting us in all the presentation. As you may have guessed, I'm not going to talk about artificial intelligence at all, but there will be some clue about innovations that we are trying to implement in our research and clinical activities in the unit of pediatric neurosurgery in our university hospital. So to introduce the topic, as you may know, in all adult tissues, which means form tissues, also in the newborn, we have distinct tissue microenvironments where somatic stem cells involved in tissue homeostasis and repair and regeneration are located. As you can see in this picture, in this scheme from this recent review, bone tissue towards the entire skeleton is provided with one of the highest regenerative potential because we have stem cell residing in it. The official and complete definition of the phenotype of these cells dates back no more than 9 years ago. It was in 2015 that they were defined as skeletal stem cells with a specific immunophenotype that you can, well, just see for your curiosity in the table. And they are, let's say, a subset of non-hematopoietic mesenchymal stromal cells, which are found in the bone marrow in most bones, not only in that. By the time they were defining mesenchymal stromal cells as skeletal stem cells in the bone marrow, we were struggling to identify, though I'm not able to use the pointer, I'm sorry, because I have a different visualization on the scheme. Anyway, in the picture that you can see on the right, we tried to systematize where are these skeletal stem cell niches located, and we ended up defining that, well, basically they are throughout the entire skeleton, and in particular, if we come to consider the differences between long and flat bones, okay, it is an oversimplification to consider that all stem cells in bones are the same. And indeed, we have different somatic stem cells residing in the skull that is formed through membranous direct ossification, and in long bones that comes from endochondral ossification. And in particular, by lineage tracing, they have been able to localize quite clearly all the different locations of stem cells in our skull, in our entire craniofacial skeletals. We have skeletal cells in all the regions that needs to form and remodel bone in the skeleton, actually. And in particular, I would like to focus your attention on what we have in our suture, where we have a, let's say, temporarily, so transient stem cell niche in the suture that will undergo ossification physiologically and, of course, pathologically in craniosynostosis. Actually, this wide heterogeneity of skeletal stem cell niches that we have in the skull may also come from the diversity by the ontogenic point of view. Because as you may know, skull bones have a mixed ontogeny. Part of the skull derived from paraxial mesoderm as most of the rest of the skeleton, actually, which is the posterior part of the skull. All the rest, which are frontal bones and all the facial bones, derive from cranial neural crests that are highly migratory and plastic cells that drive the membranous ossification of skull bones, and also, of course, coordinate the skull-brain development at the same pace, and the growth and then postnatal homeostasis of these bones. Which suggests that we have some sort of a regional segregation of stem cell niches. The first to try to categorize and describe very precisely the suture stem cells were this group in California, and they demonstrated that we have these GLI1 positive cells, so they have the positivities for this marker, which is a transcription factor of the sonic edge of pathway, and almost by the same time, we were able to originally define what we have in our bone. So the specific phenotype of skull stem cells that we have in our calvarian bones and suture niches, that they are positive for the GLI1 and the Exin2 marker. This was in 2020, and actually 2 months ago, less than 2 months ago, it was the end of September, a new paper showed up, very interesting. They do a very detailed lineage tracing study in mice where they define that not only we have separate stem cells in the skull, but within the suture we have an increased heterogeneity because we have at least these two separate population of somatic stem cells, those that are marked as CTSK positive and those that are marked as DDR2 positive. From the balance of these two different stem cell population, the current path of ossification of sutures will derive. And so basically they will be also affected in in the pathophysiology of craniosynostosis, which means that we still have a lot to study and to delve into what happens in humans with these two populations. So why do we need to have all these details from basic biology research on stem cells? Because actually stem cells can be implicated in rare diseases and in particular in craniofacial diseases, both because they can drive the pathophysiology and then they can be our target. So the target cells to be cured, let's say in our in experimental therapies, and also because we can model and study diseases by using these stem cell models. These are just a few examples that I try to gather from what we know on rare skeletal and in particular craniofacial disorders. So there are those that are due to, in the pathophysiology, they have an excessive or inappropriate bone formation, and those that instead have the contrary, the defective of ineffective bone formation. I will use craniosynostosis, so that the disease where we have excessive bone formation, because it's what I know most, of course. So in this disease, we know that we have the premature exhaustion of the stem cell residing in the suture, and we used to isolate these cells from the sutures that we obtain as surgical wastes from patient surgeries, and we base our cellular model on these cells. With these cellular model, we were able to define that these cells have a mesenchymal-like phenotype, meaning that they are the skeletal stem cells on the suture that we have defined previously. Those that are isolated from few sutures tends to ossify prematurely in vitro and tends to form bone nodules basically in cell cultures. But also we were able to demonstrate that those that prematurely ossified, that comes from few sutures niches, actually tends to express a reduced number of primary cilia. You may know that primary cilia are this sort of chemosensing and mechanosensing antenna on most somatic cells that we have in our body actually. And in the skull, they somehow sense how the extracellular matrix undergo deossification, so they increase stiffness. By having a defecting primary cilium, the cell are not able to sense the environment and undergo a premature ossification. Indeed, several genes that are known to cause syndromic craniosynostosis are involved in the function and structure of the primary cilium. Here I'm showing you this circle structure that is a multiprotein complex called BBSOM. BBS stands for Bardet-Biedl syndrome because it includes the cooperation multiple proteins that are mutated. Those genes are mutated in the Bardet-Biedl syndrome. And I'm focusing on that because these structures are relevant to form the primary cilium and to enable the signaling within the primary cilium. Very briefly, we have demonstrated that a single component, a key component of this complex, BBS9, is dysregulated in fused derived from cells derived from fused sutures. Sorry, it tends to be overexpressed and to be delocalized within the cell. Also, we have demonstrated that the expression of a specific isoform, a shorter version of this protein, is overexpressed during the course of in vivo ossification of sutures in a rat model. Talking about how to use stem cell instead for therapies, there are a lot of experimental strategies that have been exploited and are exploited in experimental therapies for tissue engineering for treating calvarial defects in skulls and also in vivo in humans and in vivo models. What I will briefly introduces the experience of our recent research in two different projects that overlap a little bit. Basically, in this project that was funded through the AFM Teleton Foundation, we developed a customized therapy that is based on targeting the allele mutated in the Crouzon syndrome, so the mutated FGFR2 allele through small interfering RNAs. My conclusion is that understanding somatic stem cells in the skull, in this case, it is can enable to improve the understanding of pathophysiology, but also to design precision medicine approach to improve therapy and provide advanced therapy in the future to assist surgery, not definitely to replace it. So thank you so much. So sorry, I have a wrong animation. Thank you so much to everyone. And this is the group of young persons that is working in the lab. Thanks.
Rhonda. Any questions there from any of our genetics colleagues in particular?
Thank you. It's a really interesting work. At what age would you recommend it, or did you do the treatment with this siRNA? Because if you want to change the disease history, you should, or are you doing it very early?
Yes, of course, we are not already in the clinical field. We are doing these days, we are starting the preclinical model in collaboration with the Laurence Leger-Mayet in Paris. So they are using this in the animal model. Of course, the time frame is different from the animal model to humans, but the idea, the translational idea is to perform this either within the surgery or right after, because using the hydrogel or either a suspension with the siRNA inside the PLGA nanoparticle, you just do a transdermal injection. So whether you have a still fused suture that is fusing in Crouzon syndrome patient that can be like without a synostosis at birth, or right after the surgery to avoid the resynostosis, you can then just simply inject. I don't know if I answered correctly your question. Okay, so it will be like in newborns.
Okay, thank you very much.